back in Stanford, I had an opportunity to work on the dynamic of design thinking with how our brain process working in a team dynamics with the concept of design thinking. Since then, I was kind of stuck with this concept of design thinking and made it kind of my day-to-day approach to solve problem, like fail fast, fail smart, fail quick. And with concept of design thinking, as the team will explain, is just you make as many prototype as possible, eliminate the one that doesn't work, and basically getting the chance of celebrating fast, fail fast, and learn through the pipeline. So I'm an AI enthusiast, and concept of drug discovery coming from bioinformatics background was one of the challenging in the field of chemoinformatics. So I was curious to see if we put a problem to any large language model and ask them to solve it, how it is changed versus when we are using the design thinking concept. So the approach was very like a bingo moment, and I kind of like it. Started looking what problem can take advantage of implementing design thinking with solving that, and that was the concept of drug discovery. So my team has implemented design thinking in a way of innovative way to use the AI enhanced framework for drug discovery. So they finished the study, they send their manuscript to a publisher, and it is under review. It's available as a preprint, which they share it with you. Here goes the team. Best of luck.
Thank you, Sahar. Hi, we're Ava, Divya, Neha, and Jennifer, and our project is Design Thinking and its application to healthcare innovation, an AI-enhanced framework for drug discovery.
So before we get into the content, we want to preface with this, that our manuscript is currently under review for the Journal of Taylor and Francis, and you can find our full manuscript preprint here at the QR code. So we'll just give you a minute to scan the QR code, and you can read this later if you're interested.
Okay, firstly, why did we choose this project? We chose to address many of the major flaws that the drug discovery process has, impacting real lives. For one, the process is incredibly slow. It takes more than 10 to 15 years for a drug to go from discovery to approval, meaning patients wait over a decade for potentially life-saving treatments. The process is also extremely expensive, over $2.6 billion to bring just one drug to the market. This limits innovation and access, especially in areas that aren't seen as profitable. Finally, the process isn't often centered around patients. It fails to meet the needs of diverse populations, leaving many people unrepresented in research and undeserved by their outcomes. All of this creates several barriers to get effective treatments to people who need them the most. Our project focuses on rethinking the system. We've explored faster, more affordable, and inclusive approaches to drug development. As previously mentioned, the drug discovery process has numerous flaws, from long timelines to high costs and a lack of inclusivity. But we believe this system can be improved. Artificial intelligence, or AI, is already helping speed up certain parts of drug development, like identifying compounds or predicting interactions. But AI alone isn't enough. That's where design thinking comes in. Design thinking is a human-centered approach to problem solving. It helps reframe challenges by focusing on the real needs of people, especially patients. Our goal is to combine these two tools. We're training large language models, or LLMs, to use the design thinking framework to solve drug discovery problems more creatively and efficiently. By doing this, we aim to create a system that's not just faster and smarter, but one that truly prioritizes patient-centered care.
Design thinking has been previously applied in healthcare settings to improve patient care and redesign system processes. By offering a human-centered approach to reframing issues in healthcare organization, design thinking allows researchers to focus on efficiency and patient care. For example, by using design thinking, an emergency department implemented patient advocates to open up communication between providers and patients. This led to greater patient satisfaction and efficient wait times. Design thinking in high-stress areas like the ICU has also led healthcare innovators to target operational inefficiencies, such as redesigning layouts of medical devices and equipment for optimized patient care. Another example of design thinking is its application in radiology. By analyzing patient needs, innovators were able to redesign X-ray spaces to create a less intimidating atmosphere. By using design thinking to reframe healthcare issues, innovators are able to focus on patient needs and understand the overall perspective of the issue. So one of the things that we looked into while narrowing down the scope of our project was, of course, the drug development stages and timeline. The first part of drug development is conducted outside of humans, where the drug is actually discovered and developed. This process takes several years and has incredibly high R&D costs. Many drugs move on through this process but fail in later clinical trials, specifically phases 2 and 3 of human trials. So this is after a lot of time, money, and effort has already been invested by labs and research facilities, making drug development a highly inefficient process as it stands today.
Next, moving on to the background and what is design thinking. Design thinking is a 5-step process to solve problems a bit faster. The 5 stages of design thinking are empathize, define, ideate, prototype, and implement. The first stage, or empathize, is to understand the audience's perspective. So for example, in our situation, that would be understanding that different people have differences in drug dosage requirements. So basically understanding the different needs of different people. The next stage, the second stage, is to define, which is crafting a problem statement. For example, in our situation, that would be creating personalized drugs that fit into individuals' needs. The third stage is ideate, which is when people produce as many ideas as possible, or rapid iteration. For example, this is the AI tool putting all drugs into one capsule, or the AI tool that scans for personalized drug dosages. So pretty much all the possible solutions that the AI can think of. The fourth stage is prototype, or creating a rough representation of the best ideas. This could be creating a sample model of the ideate stage. The final stage is implement, which is where we test our prototype and gather feedback on it. For example, this could be testing our product on a small group of people. Through the design thinking process, solutions are able to be created much faster.
So before we dive into the results of our study, it was really important for us to first compare the capabilities of different large language models, or LLMs, and explore why testing multiple models matter in pharmaceutical research. We wanted to identify which model performed best at different stages in the drug discovery pipeline, from ideation to prototype. Each model has unique strengths. For example, some simplify complex data better, while others generate novel solutions. Incorporating these models into a design thinking framework would allow us to evaluate their both creative and analytical potential. More importantly, we want to ensure that the responses that they give aren't just accurate, but also ethical, explainable, and centered around real patient needs. So let's briefly break down what each model brought to the table. This is based on previous literature reviews and past usage. ChatGPT excelled in structured content, handling complex prompts and generating detailed research explanations. It was particularly strong for tasks like scientific rationales or refining technical content. ChatGPT is also customizable, though we didn't use this feature through APIs and plugins. Gemini, on the other hand, stood out with its ability to process text, images, and audio, as well as video natively, which also makes it ideal for when we're working with multimodal drug datasets. It consistently gave us research-backed response and simplified technical concepts well. Lastly, DeepSeek impresses us with its speed. It's ideal for quickly generating long-form drafts or even early-stage content. However, DeepSeek is usually seen to have less variation in responses, and it has limited source transparency, which we also know as a trade-off for its efficiency. This general model comparison gave us a strong foundation for integrating the AI tools when we eventually conducted our experiments later.
So throughout our project, we narrowed down the problems that we're focusing on into 5 different problems. The first is with QSAR, which is quantitative structure activity relationship modeling. This is basically a computational method which uses artificial intelligence in drug discovery to enhance molecular design, drug repurposing, and toxicity prediction. This method can model how a drug might interact with the target, but it often struggles with issues like overfitting and predicting accidental interactions with other proteins. It might also have high costs, time consumption, and low efficacy, which requires much improvement before it can be used regularly for drug design. The next is de novo molecular design, which involves generating novel chemical structures that meet specific objectives such as biological effectiveness, safety, and synthesis feasibility. A key challenge with this one is balancing all these factors, as highly effective models may be difficult to synthesize or pose safety risks, while simpler molecules may lack potency. While generative models and computational methods can explore chemical space more efficiently than traditional approaches, they still struggle to predict the real-world feasibility of molecules. As a result, de novo design often fails to generate compounds that optimize these trade-offs, limiting its effectiveness in drug discovery. The next is organ on chips, which was designed as an alternative for animal testing. Animal testing procedures have very high failure rates, are often inaccurate representations of humans, and have ethical concerns. In the organ on chips model, cultures of engineered miniature human tissue systems are developed in microfluidic chips. This was formed to create an accurate model to test drugs on instead of using animals to test treatments. However, this model has a lot of room for improvement, with the main issue being that when used for drug development, it has difficulty in fully replicating the complexity of human physiology, including interorgan interactions, immune responses, and disease-specific factors, while also ensuring scalability, reproducibility, and regulatory acceptance. So the next two that we focused on are problems with iPSCs and druggable protein targets. iPSCs, which is short for induced pluripotent stem cells, are somatic cells that are reprogrammed to their pluripotent state and can then be differentiated into various cell types that model human diseases. In vitro cell-based models of disease such as this one hold promise for monitoring disease progression and can improve drug testing on patient-derived cells, limiting unexpected side effects. However, the potential of iPSCs is unstable and limits their effectiveness for clinical and research applications. They have unpredictable changes in their DNA and gene regulation during or after reprogramming, which then leads to reduced functionality. And so then lastly, we have protein targets or druggable protein targets, or like the lack thereof. So protein targets are only considered druggable if they can bind to small drug-like molecules and produce a desired therapeutic effect. It's considered undruggable if it is difficult to target for drug development. So some contributing factors that might make a protein target undruggable is if it has flat functional interfaces without defined pockets for ligand interactions, it's involved in several other protein-protein interactions, or it's an intracellular protein rather than an extracellular protein. As of today, about 85% of proteins are considered undruggable, which translates to about 60% of drug discovery projects failing due to undruggable targets.
Next on to a bit of methodology for our project. So as you can see, our project consisted of multiple phases. The first of which was learning about the design thinking model. This involves the pill bottle model you saw a few slides ago. In this stage, we conducted a lot of literature reviews, looking into a lot of different sources and how they describe the design thinking process. The next stage was applying design thinking to common problems. So this included trying to apply design thinking to problems like missing socks in the washing machine or hangers breaking over time. So through applying it to these different problems, we were able to further understand how the design thinking process works. The third stage was literature reviews, and specifically literature reviews of design thinking in healthcare. Based on this, we were able to see how design thinking has previously been used in healthcare and what potential there is to further incorporate design thinking into healthcare. The next stage was analyzing human versus AI approach to problem solving with design thinking. So this involved plugging the same problems of like the breaking hangers and the missing socks into AI using design thinking and seeing how they solve the problems and comparing the similarities and differences between the human approach and the AI approach. The next step is a literature review of the drug discovery pipeline. This involved researching all the different issues with the drug discovery pipeline, including high costs and low time efficiency. The next stage was problem identification of the drug discovery pipeline. So this involved finding all of the different bad parts of the drug discovery pipeline and how they could be fixed. The next step was a qualitative approach and plugging drug discovery problems into AI. So this includes plugging the six different problems previously mentioned into AI using design thinking and seeing how it solved it, with and without design thinking, creating a comparison here.
So now that we've given some background information and shared some goals of our project, now let's move on to talking about the data collection process that we went through. So to give an overview, first, we prompted the AI using a reference abstract to define key challenges in each problem. And that's how we were able to define shortcomings in the different topics that we introduced previously. Then we prompted AI to ideate solutions to each problem without using the design thinking framework. And then we prompted it to reapproach the same problem using the design thinking framework. Then after that, when we collected data from all three LLMs using all of our prompts, we were able to evaluate them, kind of note differences, do a comparison, and analyze the results that we got. So to give a sample response of something that we looked at is this table. So this is like the first half of it. For our paper itself, we included like a few sample responses like this, but the rest of them we included in a secondary material because there was just like way too much to include, and this is just like one of the examples of such a response. So this is ChatGPT's response to problem number 1, which is the organ on chips problem. And as you can see, there's the problem category, which is organ on chips, then the prompt where we prompted it with an issue pertaining to the specific topic. Then we included the response that was given with design thinking. So as you can see, it's following like this shows the ideation phase because the entire response was too long. But like that's generally the part we're looking at to look at the different ideas it comes up with. So that's what it gives here. Then we included the response without design thinking. So after we collected all of these results, we were able to kind of look at it as a bigger picture and compare and contrast them. So to give a kind of quick visual overview of what we got here. So first we did like a human and AI comparison, you know, with the human examples that we looked at before we dove into the AI part. So we concluded that humans were more creative, but AI was broader. Humans were more specific to the user, whereas AI was more detached from the problem, things like that. And then we did a cross comparison of the 3 different LLMs that we were comparing through our study, which we will be going into more detail in a second, but broadly, ChatGPT, as we mentioned before, provided more info, whereas Gemini had broader solutions and DeepSeek was more user-centric. But overall, there are a lot of more nuanced comparisons that we will be going into in a second.
After an evaluation of our results, we need to discuss how we balance the strengths of AI that we saw with the value of human reasoning through the design thinking process. First, AI gives us incredible speed. Processing huge data sets, it can spot patterns and optimize even predictions. But what it lacks is context, the ethical and human understanding that is needed in medicine. That's where human cognition through design thinking plays a critical role. Our creativity, our ethics, our real world experience fills in those gaps. Design thinking bridges this divide. It adds structure and empathy to the process. Empathy is the first step of the design thinking process. And so instead of a rigid linear approach, it allows for an iterative, refined, and rapid testing, along with constant feedback that is especially useful in a field that is as high stakes as pharmaceutical and drug development. This framework also shifts the focus from the molecule, the drug, to the actual person and the patient. Whether it's accounting for genetic diversity in personalized medicine or minimizing animal testing by improving biological relevance that we saw in that problem. The combination of AI and human centered design unlocks new opportunities for innovation. Our research also looked at the systematic challenges in drug discovery today. To review, on average, it takes over 10 years and $2 billion to bring just one drug to market, and only a small percentage gain FDA approval. Worse, most trials don't reflect diverse populations, which results in higher rates of adverse drug reactions, especially in minorities and women. As patients age, they're often prescribed multiple medications, and when those drugs aren't tailored to them, risks only increase. This specifically is where AI and design thinking make a real difference. AI speeds up development, while design thinking ensures what's developed is safe, inclusive, and relevant. By combining the precision of AI with empathy of human centered design, we can also reduce trial and error inefficiencies and move towards a pharmaceutical system that truly works for each patient. To expand on the individual models that we tested, in summary, we tested 3 models. Again, ChatGPT, Gemini, and DeepSeek. And each one had different strengths. We knew this going forward. So ChatGPT emerged as the most well-rounded performer. It consistently generated detailed and structured content that aligned closely with scientific language and also context in a pharmaceutical industrial setting. During empathize and ideate phases of design thinking, where the human portion really comes out, it responded well to refined prompts, and it was also able to adapt its tone and level of complexity based on patient needs or based on the individual problems that it was solving. It also offered a creative yet practical solution, and it struck a strong balance between innovation and technical accuracy. Gemini had strong capabilities in other areas because it stood out with being able to work with our big paragraphs of text that we were inputting into it, and it simplified complex medical concepts effectively. It provided source backed information, which is crucial for real world credibility. However, we saw that Gemini also lacked the same depth and iterative ideation. It did give solid drafts, but it didn't evolve responses as effectively. Finally, DeepSeek was best for speed. It was able to generate long-form content quickly and was used for rapidly generating ideas. But as well, it lacked variation and offered fewer nuanced insights that we saw, especially when tackling problems that require more ethical or patient-centered thinking. So overall, ChatGPT paired with design thinking delivered the most dynamic, human-centered, and feasible responses. It was the most responsive to our prompt engineering processes, and it also consistently produced ideas that were both adaptable, inclusive, as well as aligned with the realities in the pharmaceutical industry. That's why for future pharmaceutical design workflows, we recommend prioritizing ChatGPT when building LLM enhanced solutions that centered the needs of diverse patient populations while maintaining scientific accuracy. This brings us to the end of our presentation. Thank you so much for listening, and we would like to acknowledge Sahar Jahanikia, our principal investigator, for guiding us through this process, as well as ASDRP admin and the Olive Children Foundation for helping us. And we now open the floor to any questions.